Hello everyone, welcome to this lecture in molecular biology where we'll start chapter 5 covering molecular tools for studying genes and gene activity. The topics we'll discuss in this lecture are methods to separate molecules like nucleic acids and proteins, labeled tracers to detect nucleic acids and proteins, and nucleic acid hybridization techniques to detect specific sequences. These are covered in sections 5.1 to 5.3. For molecular separations, we'll focus on electrophoresis of DNA, RNA, and proteins, and a variety of chromatography methods to separate different types of molecules. First, we'll talk about DNA electrophoresis, which is typically performed in an agarose gel. In any kind of electrophoresis, you have cations, or positive ions, going to the cathode, the negative electrode, and anions, or negative ions, going to the anode, the positive electrode. In DNA electrophoresis, is DNA a cation or an anion? Is it positively charged or is it negatively charged? DNA is negatively charged. It's an anion. The negatively charged DNA goes to the positive electrode. Where does the negative charge on DNA come from, by the way? It comes from the phosphate group. Nucleotides within nucleic acids are linked by phosphodiester bonds, and that's where the negative charge comes from. Each nucleotide on DNA carries a negative charge on the phosphate, so you have an essentially uniform negative charge on a DNA molecule. The negatively charged DNA migrates to the positively charged anode. The uniform charge on DNA means that the total charge is proportional to the size of the DNA, and you have a uniform mass-to-charge ratio on DNA. So, everything else being equal, small and large DNA molecules would migrate at the same rate, but in an agarose gel, the larger molecules experience more drag than smaller molecules as they travel through the gel, and so the larger molecules move slower. In this way, you can separate DNA molecules by size when you run these gels. You can visualize the DNA by using a dye, such as ethidium bromide, that becomes fluorescent when it binds to DNA. You can see that if you illuminate the dye under UV light, you see sort of a pinkish fluorescence where it's bound to DNA. You can also purify DNA molecules that you separate in this way. You can cut them out of the gel and isolate the DNA. The electrophoretic mobility, which is to say the distance that the DNA will migrate during electrophoresis, has a relationship to its molecular weight. It's not a direct linear relationship to the size of the DNA molecule, but it's a linear relationship to the log of the molecular weight. You can see here that if the DNA is migrating from the top of the gel, the larger molecules migrate less. You can plot this on a log graph and you will see that the distance it's migrated is proportional to the log of the molecular weight and not linearly proportional to the molecular weight itself. Using this relationship, you can obtain a standard curve by plotting the mobility of DNA fragments of known molecular weight in order to determine the weight of an unknown DNA sample by comparison with those standards. That's the normal, straightforward application of DNA electrophoresis. Normally, that sort of electrophoresis is done with a constant electric field in one direction. However, when carrying out DNA electrophoresis in this fashion, the relationship between the mobility and size breaks down for very large DNA molecules. Above a certain threshold, these large DNA molecules will all appear to migrate at the same rate, so you won't be able to distinguish the size of these large molecules beyond that threshold. In order to distinguish between the sizes of very large DNA molecules using electrophoresis, a specialized method called pulsed gel electrophoresis is used. This operates by periodically changing the direction of the electric field. The result is that these large fragments of DNA that are above the normal threshold for normal gel electrophoresis, they will reorient to the changing direction of the field at different rates, and that will affect their ability to move through the gel, and you can distinguish them on that basis. What do I mean by changing the direction of the electric field? Normally, you do the electrophoresis with a constant field in one direction, for example, oriented so that the DNA goes down the gel. In this pulsed gel electrophoresis, you may have a pulse where you do that, then you'll have a pulse where you change the direction of the field at sort of an angle relative to the direction that you want the DNA to go, and you can have several of these pulses so that the net direction that these DNA molecules are going is still down the gel. By having a series of pulses in which you change the direction of the field, the larger molecules need more time to reorient before continuing to move through the gel in the electrophoresis. This is how you can separate very large DNA molecules, from tens of thousands of base pairs and going higher than 1,000,000 base pairs. That's DNA electrophoresis. You can do electrophoresis on proteins as well. This is usually done in a process called SDS page. So how would this work? Do proteins carry a charge? Are they positively charged or negatively charged? Well, it depends. It depends on the protein sequence and it depends on the pH. At a given pH, a protein might be more positively charged or more negatively charged compared to at a different pH. We'll touch on that more in a little bit, but still you can use electrophoresis to separate proteins by size. How do we do that? If you have a protein, typically it's folded up, and it might have a positive or a negative charge, or it might be net neutral. But if you apply this detergent, sodium dodecyl sulfate, or SDS, which is illustrated here, you can confer a uniform negative charge to the protein based on its size. SDS has a charged polar head group and a lipid tail. You can use the detergent to essentially unfold the protein into a linear chain by disrupting the interactions keeping the protein folded. The hydrophobic lipid tails of SDS bind nonpolar regions and disrupt the hydrophobic interactions mostly in the core of the folded protein, and the polar head groups disrupt the polar interactions that help to fold the protein. So you end up with an unfolded protein that is evenly coated with detergent molecules, which confer a negative charge so that the charge is uniform on the unfolded protein. So, the bigger the protein, the more detergent is interacting with it, and the more negative charge is on that complex. But the mass to charge ratio remains the same. You can separate them by size following the same principles using electrophoresis, because the larger proteins experience more drag in the gel. That's illustrated here in this image. In this figure though, these proteins have been pre-stained with colored dye before they've been denatured by SDS and before they have been separated by polyacrylamide gel electrophoresis, or PAGE, SDS PAGE. So you can see them as different colors. Normally, if you're analyzing an unknown sample, you run the proteins in SDS PAGE and then you stain them afterwards with a dye. When you wash away the dye, the proteins will remain stained, and you can visualize the proteins based on that staining. So that's useful for separating and analyzing polypeptides by size, but what if you have many proteins and polypeptides in a complex sample? Maybe a whole cell lysate from some tissue or culture. There are other specific methods of gel electrophoresis that you can use, such as 2D gel electrophoresis, that allow you to resolve complex mixtures of polypeptides, thousands of polypeptides for example. These can be separated not only by size but by other properties as well. A typical 2D gel electrophoresis separates proteins by size and also by their charge at a given pH. As I'd said before, natively, different proteins will have different charges, and this also depends on the pH. The charge on proteins will depend on the acidic and basic residues on that protein. Acidic residues at low pH may be neutral, but at high pH they may be deprotonated and carry a negative charge. Basic residues at low pH may be protonated and carry a positive charge, but at high pH they will be neutral. Proteins can be separated on this basis if you perform electrophoresis on them in a pH gradient. So you can have low pH here and high pH there, and put your anode here and your cathode there. At a high pH, a protein might have a negative charge, and it would be attracted to the positive electrode, the anode, and repelled by the cathode, so that it moves along this gradient, causing its charge to become less negative as the pH is changing, until the net charge on the protein becomes neutral. Same thing on this end. You might have the protein positively charged at lower pH. It is attracted to the negative electrode and repelled by the positive electrode, causing it to migrate along the pH gradient until its net charge becomes net neutral. At a certain pH, a given protein will have a net neutral charge, and using this technique, you can cause a protein to settle to a position in the pH gradient where its charge is net neutral. This is called isoelectric focusing. You can separate proteins in one dimension using isoelectric focusing, and in the second dimension, you can do SDS-Page and separate them by size. The result is you get a complex pattern of spots. Shown here on this example, you have two protein samples that are coming from E. coli. The cells were grown in different conditions. One was grown with benzoic acid in the culture medium, and the other without. Each of the protein extracts were separated on a 2D gel and stained with a different dye. These are fluorescent dyes, Cy3 or Cy5, for cells grown in the absence or presence of benzoic acid, respectively. You can overlay the resulting 2D gels and you can pick out which ones are different. So these spots in red that don't correspond with a blue spot are proteins that are expressed in the absence of benzoic acid, but not in its presence. And the spots in blue that do not correspond with red spots are proteins that are expressed in the presence of benzoic acid, but not in its absence. Having covered electrophoresis, now we'll get into other molecular separation methods. Here we will discuss chromatography. There are three examples of chromatography we'll talk about. We have ion exchange, gel filtration, and affinity chromatography. In ion exchange, we're separating molecules based on charge. A molecule, for example a protein, might have a net charge at a given pH. It will bind to a positively or negatively charged resin, depending on whether it is negatively or positively charged, and you can elute that protein by gradually increasing the salt concentration, which increases the ionic strength. Shown here on this slide is one example of that from your textbook. So how this works is you have a column containing a resin, and let's say this resin is positively charged. And you have your protein. Your protein sample is buffered at a given pH. At that pH, the protein is negatively charged. The negatively charged protein binds to the positively charged resin. Then, to elute your protein, that is, to collect it from the column, you gradually increase the salt concentration. If you follow the example in your textbook corresponding to this figure, you add potassium chloride. Then negatively charged chloride anions exchange with your anionic protein, and then your protein is eluted out. That's why this is called ion exchange. This was an example of anion exchange. If you have the reverse situation, a negatively charged resin and a positively charged protein or molecule, that would be cation exchange. Another type of chromatography we will discuss is gel filtration. This separates molecules by size. You might have a column with a resin that has pores within it. So there are holes and spaces inside the beads that make up this resin. This is useful if you want to separate molecules of different size. Some small molecules will be able to fit inside these pores and travel inside these beads. Larger molecules will be excluded from the pores and won't be able to travel inside these beads, and they'll instead have to travel around the beads. Effectively, what happens is that the smaller molecules have more space to travel in, whereas the larger molecules have less space to travel through. They go through what's called the void volume. That's the volume that's not within these resin beads. So they effectively are traveling through less volume, so as you are flowing buffer through this column, less volume is needed to elute these large molecules through the column than the small molecules, because the small molecules are moving into these beads and floating around before they can get out of the column. This is the basis by which gel filtration works. Large molecules are excluded, small molecules go through all the space, and intermediately sized molecules might get into some of these larger pores and not smaller pores, so you can get a separation of molecules according to size. Here on this slide, we discussed ion exchange and gel filtration, and we already talked about affinity chromatography with an example of his-tagged proteins. Affinity chromatography is based on specific interactions. His-tagged proteins associating to immobilized metal ions is one example. You can have other examples of specific interactions being used to separate molecules, for example, using antibodies. Now that we've covered molecular separations, I will pause here before getting into the next part of the lecture, talking about labeled tracers and nucleic acid hybridization techniques.